This is an educational video that focuses on immunogenic responses to drugs and specifically on anti-drug antibody formation and isotype switch. Through the talk, I will be going over 6 questions and I will explain the answers to these questions. They are posed as statements that are either true or false. So you can pause the video right now, you can take a screenshot and have an attempt to respond in the questions before we move on. Or you can just choose to go on with the video and see the responses as we go along.
Immunogenicity is an idiosyncratic response to a drug. That's a therapeutic protein. What happens is that the particular drug will be perceived as a danger signal by the B cell or the dendritic cell. Both of them are antigen-presenting cells. They can basically process the whole drug that is perceived as a danger signal and actually present, that's why they're called antigen-presenting cells, they present the T cell epitope that then the T cell will recognize as dangerous, and in the appropriate conditions of co-stimulation, this T cell will be activated to mount the immune response against the drug. Basically, this T cell response will produce certain cytokines that can activate eosinophils or neutrophils or macrophages. And these would be the cells that would be dealing with getting rid of that danger signal, because that's the whole point of the immune response. On the other hand, the T cell response can trigger a further B cell response with the appropriate cytokine stimulation. It activates B cells and makes them mature into plasma cells. It is the plasma cell that is responsible for the anti-drug antibody induction. So basically, the anti-drug antibodies are going to bind the whole drug, not just the T cell epitope. And the eosinophil, neutrophil, and macrophage will also be dealing with the whole drug as a danger signal, not just the T cell epitope. But it is the T cell response, the one that triggers this whole and orchestrates, let's say, this whole response to get rid of something that is perceived as dangerous. It is also the T cell response, the one that could produce memory B cells. These cells will now identify the whole drug and they will be able to respond faster with future exposure to the drug. This immunogenicity is an unwanted effect of the therapeutic proteins. We definitely do not want the immune system to mount an immune response against the drug, because it would decrease efficacy and it can cause safety concerns. The only area of development in the drug development work where we actually desire an immunogenic response is vaccines, where the whole point is that what we are vaccinating with, we are expecting to mount an immune response that will be protective for the individual. But in all the other therapeutic protein examples, immunogenicity is not desired. Immunogenicity is an idiosyncratic response. What this means is that different individuals will react differently. While we know that some danger signals are identified by everybody, like crystals or cell walls and pathogens, because they're not normally seen by our body as part of ourselves, there are other cases and ample examples of different antigens in nature that are seen by some individuals as dangerous, like some people have poison ivy allergy or allergy to peanuts. And this is not everybody, it's idiosyncratic. We also react to other individuals, to other people's antigens in different ways. Sometimes the reaction could be very dramatic. And this is evidently something that we need to match before a tissue transplant, an organ transplant, or a blood transfusion, for instance. So this is unique. And we need to understand if a particular individual is going to react to a drug as if it was a danger signal. This is really difficult to predict, and we can only measure it once we're doing the drug development. If an individual does mount an immune response against the drug, this particular individual can have the T cell response or the B cell response or both. And this anti-drug antibody induction is a very useful tool that we have to measure anti-drug antibodies during clinical trials to characterize the frequency by which a particular drug is perceived as dangerous and is mounting an immunogenic response to the drug and during the clinical trial development. Not all of them are going to produce memory, not all of them are going to produce anti-drug antibodies. Some of them are going to have only a T cell response, others a B cell response. But in all cases, it's idiosyncratic. We do need to understand if an individual reacts or doesn't react to that drug, if that reaction is creating a memory to it, and also how intense and how severe this reaction is so that we can treat on a case-by-case basis.
All right, so let's see if these first three statements are true or false. Immunogenicity is an idiosyncratic, unwanted immune response to any drug that is perceived as danger signal by the immune system. The key why this is false is unwanted because, as I explained briefly, in vaccines, that's actually the desired effect. So if you do not take into account vaccines, yes, this is unwanted for all the other drugs. And it is idiosyncratic. Presentation of T cell epitope to the T cell receptor in the presence of co-stimulatory signals triggers Th differentiation that leads to antibody formation. That is true. It is a Th differentiation that leads to a B cell activation and maturation. So without the T cell, the B cell will not be able to produce the mature response in the form of anti-drug antibodies. Immunogenicity is a response attempting to get rid of a drug by the effector cells, Th1 or Th17 driven, and or by the presence of anti-drug antibodies, which is Th2 driven. This is true. So the immunogenicity is not solely anti-drug antibody formation. In some cases, we could have activation of macrophages or neutrophils or eosinophils that will be driving a cellular immunogenic response in the absence of anti-drug antibodies. So focusing on immunogenicity, let's take a look at the B cell response. When a drug is perceived as a danger signal by the dendritic cell, it can process the drug and identify and digest that T cell epitope, the epitope that the T cell can see as danger signal. Likewise, the B cells can also identify the whole drug as a potential danger signal and also process the same T cell epitope. These two cells have the ability to present the epitope to the T cell. That's why they are called antigen-presenting cells. And the T cell will recognize that epitope through the T cell receptor. This engagement from the antigen-presenting cells to the T cell via the T cell receptor with the appropriate co-stimulatory factors will differentiate this T cell into a Th2, producing interleukin-4 that will then activate and maturate the B cells into plasma cells. Plasma cells are the cells that will secrete the anti-drug antibodies. These antibodies can be IgM, IgG, IgA, or IgE, but all of them independently of the isotype or also known as the antibody class, all of them will bind the original drug, the same drug that triggered all of these events. So in the same individual, we can find different isotypes, but they all have the capability of binding the whole drug. This whole process is triggered by the Th cell, and specifically the Th2 is a phenotype that allows this differentiation and maturation of the B cell lineage into producing antidrug antibodies. In addition, this Th2 cell can also stimulate these B cells producing memory B cells. These memory B cells now have the ability to identify the original drug. Not everybody who has an immunogenicity B cell response with the presence of antidrug antibodies has memory to them. Sometimes the process occurs, they will have a first stimulation differentiation into plasma cells. These plasma cells could be short-lived, and it is seen in some cases that antidrug antibodies could disappear on time, even though the patient could be receiving the drug repeatedly. This has to be assessed on a case-by-case basis because it is idiosyncratic. So when we have antidrug antibodies of different isotypes, this means that there has been a stimulation on the B cell. Let's go over the process for isotype switching. We have the naive B cell that is responding to a drug that is perceived as antigenic, as a danger signal. In the germline gene of the B cell, we have the variable region. This is the one that's identifying the antigen. But moving to the right, we have the different genes for the switching regions and the constant regions. The capital S identifies a switching gene, and the capital C identifies the constant region. The Greek letter mu identifies the IgM gene, so it's a switch for IgM, and the constant region for IgM. We have the delta for IgD, we have the gamma for IgG, and the epsilon for IgE, and then we have alpha for IgA, which is not shown in this particular cartoon. The order goes to the right from mu and delta, which are the first ones, all the way to eventually IgA. What makes a B cell that originally will be producing IgM, which is the first gene it encounters when it first secretes the first antibody? What makes this B cell change the isotype? It all has to do with the stimulation from the B cell that presents the antigen to the T cell via the T cell receptor with the appropriate co-stimulatory factors. And this Th cell, first at Th2, differentiates the original B cell and matures them into plasma cell, but a continuous engagement with T cells can differentiate them into Th1s, Th17s, besides the Th2, and they can drive the different isotype switching on the mature B cells. In other words, the signals from the Th cells with the co-stimulation via the CD40 ligand and the specific secretion of cytokines by those Th cells is what's going to help identify which is the germline transcript, for instance, if we are going to be switching to an IgE. And all this time, the variable region still remains identifying the drug.
Let's take a look at the adaptive immune response Th cell phenotypes. When we have a naive T cell that is actually presented a particular T cell epitope by the dendritic cell with the appropriate co-stimulation and a particular interaction with the production of interleukin 12 or interferon gamma will differentiate that naive T cell into what we know as a Th1 cell. This Th1 phenotype is characterized by the production of several cytokines such as TNF-alpha, interleukin-2, interferon gamma, and interleukin-18. This is important because these cytokines target a key effector cell that is called macrophage. The macrophages will be associated with a Th1 response. They are activated by these Th1 cells, and they deal with intracellular pathogens, with tissue repair, foreign bodies. So this is the most common inflammatory response that we have, dealing with the different danger signals, including, for instance, when we cut ourselves, or even when we have a burn, when we burn ourselves, or foreign bodies, like splinter or intracellular pathogens, which is very well known. The mechanism by which the macrophages deal with this danger signal is driven by the interaction from the dendritic cell to the co-stimulatory cell. That original interaction is going to make sure that this phenotype is the Th1, and that these macrophages will be there activated, ready to produce either phagocytosis, direct killing of those intracellular pathogens, or oftentimes when they cannot actually kill the pathogen, they could also produce a granuloma formation either around the foreign body, or in the case of tuberculosis, the bacillum. This granuloma will not allow that danger signal to spread. So there's different ways that macrophages defend our bodies, and this is all driven by that T helper phenotype Th1. This is also a mechanism that plays an important role in hypersensitivity type 4 to either different kinds of foreign antigens, or it could also be another foreign antigen, could be a drug. So immunogenicity, that's cellular immunogenicity, could be resulting from macrophage activation against the drug when this particular dendritic cell presents the drug as a signal that is dangerous. In another interaction with different co-stimulatory factors and different T-cell receptors to those epitopes, the interleukin-4 environment will now differentiate the T helper cell into a Th2 phenotype with a characteristic production of cytokines that include interleukin-4, interleukin-5, interleukin-13, and interleukin-25. Note that the cytokines produced by different phenotypes of the T cell do not overlap. They're very characteristic and different. These cytokines will in turn stimulate basically eosinophils. This Th2 response is the way the body has differentiated the response to deal with parasites, allergens, and drugs as antigens. And the typical mechanism will be atopic, mediated by the production of IgE, or it could be antibody mediated by IgG or IgM. It can also cause mast cell degranulation, and it is responsible for hypersensitivity types 1, 2, and 3. Finally, the other key phenotype for this response would be when the interaction between the co-stimulation and the particular T cell receptor that is engaged by the epitope produces either interleukin-1, interleukin-6, interleukin-21, or interleukin-23, all of them in the presence of TGF-beta. This is the situation where a cocktail or a combination of cytokines with the mandatory presence of TGF-beta with this stimulation will differentiate the T cell into a Th17. And the cytokines that characterize the Th17 are the interleukin-17 cytokines, interleukin-21, and also TNF-alpha. In this case, the target cell is a neutrophil. So what is that completes the associated response? Well, the Th17 response typically deals with extracellular pathogens, like fungi infections. So we now have completed the typical responses that we need with 3 different Th phenotypes. And the mechanism of the Th17 is basically production of antimicrobial peptides, inflammatory reactions, the barrier integrity, for instance, from the neutrophils on the skin, or the GI tract, or the respiratory tract is very, very important to maintain the danger signals from causing disease. This is also one of the potential mechanisms for hypersensitivity type 4. So hypersensitivity type 4, which is also called delayed type hypersensitivity, it could be either a Th1 or a Th17 response. Hypersensitivities types 1, 2, 3 are, on the other hand, Th2 responses.
So the immunogenic B-cell response characterized by the presence of antibodies to the drug requires that the drug be identified by antigen-presenting cells, mostly dendritic cells, but the B-cell is also involved, that, as their name implies, present the antigen to the T-cell. This T-cell with the appropriate co-stimulatory factor will originally be differentiated into a Th2 with the secretion of interleukin-4 that eventually matures and differentiates the B-cell lineage into a plasma cell, which is responsible for the first IgM, antidrug antibody. What causes the switch? Well, not all of the cases of antidrug antibodies will have an isotype switch. Sometimes they will remain being IgM. What will determine an isotype switch depends on a continuous stimulation from the antigen-presenting cells to the T cell that now can differentiate into different phenotypes and produce different kinds of cytokines, and this pattern of cytokine secretion will then stimulate the memory cell into rearranging the heavy chain genes that will switch from an IgM all the way to IgE. So once the naive B cell has identified the drug as an antigen, it somatically mutates the variable region to bind that antigen, but we're going to be focusing now on this talk on the isotype switch. The genetic sequence for the isotypes or the immunoglobulin classes are always ordered from IgM to the different isotypes downstream. The direction is always the same. It always goes from the IgM all the way to IgA2. This is from human sequences. You are going to find several different publications on rodents. And what I decided to do is to look at several publications and gather all the information and summarize it as far as I can understand it in this particular talk. So this is the order that I have seen published for the human genome. What I also put in here was a little oval before the rectangle. The oval signifies the switching region, the gene for the switch. If you notice, the IgD construct does not have a switch. This explains why the B cells that are immature B cells, they first produce the IgM because it's the first construct they find, but they can co-express IgM and IgD. Both of these will be binding the same antigen, but they can be found on the same cell. However, once they switch isotype further downstream to the right, then you don't see co-expression or co-secretion of the same isotype of different isotypes by the same cell. Let's go over this process so that you understand better how this happens. So it is the signals from Th cells that have been stimulated through the T cell receptor by the specific drug that is perceived as antigenic. So it's the T cell epitope from the drug, and with the appropriate co-stimulatory signals from the CD40 ligand that produces a different pattern of cytokines. And these are the drivers for the switch. For the switch, the B cell actually can identify the switching regions, and they actually bind them together, splicing out the rest of the genome, literally cut it out. So once a cell has switched to a particular isotype gene, using the switching regions as identifiers for that binding, it splices out everything else so it can never go back in the other direction. So it always goes on the direction from left to right, as I showed in the cartoon. By splicing out that gene, now the B cell has a recombination from the two switched regions, and now the construct in this example is for the constant region epsilon. This will allow the cell to be producing a transcription to a messenger RNA for epsilon, which translates into a protein that's IgE. Again, the variable region has not changed in this example. We're just showing how the same identifier for the same drug will now become an IgE by splicing out the genes, intermediate genes, and selecting the IgE. And this is driven by the specific Th phenotype and the cytokines that were stimulated by that first interaction or sustained interaction from the drug epitope being presented to the T cell. Now, what are these cytokines? Again, there's many publications. I encourage you, if you're interested in the field, to take a look at all of these publications. Some of them are really old, some of them are more recent, and this characterization has a lot of different details that you need to take into account. Number one, I decided to just look at the results when they used human B cells. There's a lot of information on rodents, and we do share a lot of different pathways, but I prefer to focus on what I could find from human B cells. Second, these experiments have to be done always in vitro with the isolated B cells, and the conditions for isolating the B cells and culturing them and activating them, the technology for measuring activation and measuring the response to a particular isotype change over time. So that's why we can find some publications missing one or another signal, and some others could have signals that are yet to be described. So what I found was basically, as an example, Th2-driven response that secretes interleukin-4 will favor isotype switching to IgG1, 2, 4, and IgE. The interleukin-5 secretion by a Th2 phenotype will actually stimulate the switch to IgA. So this is an example of how the Th is really orchestrating the whole response, not only identifying the antigen, but which isotype has to be switched to. And the isotypes have different characteristics and features for dealing with what's perceived as the danger signal. Interferon gamma will stimulate IgG3 switch, while TGF-beta will stimulate the switch to IgA and interleukin-10 to IgG1 and IgG3. On the other hand, there's publications that seem to suggest that interleukin-4, for instance, can inhibit the switch to IgG3. Interferon gamma inhibit IgG1 and IgE switch, and TGF-beta inhibits the switch to IgG3. This is not final. As I mentioned, there's many new technologies, many new assays, and many new cytokines that are described. But this is just an example so that you can understand better how the different drugs can be seen as immunogenic, and this really depends on each individual, how these T cells are going to react to that epitope presentation, and how they are going to be producing different cytokines. And this is why it is idiosyncratic. And it can also change with time. An individual may not always produce IgE in response to the same drug, for instance, when we're talking about immunogenicity. This individual, especially if there's an IL-4 driven response, this individual could actually have some cells switching to IgG1, some others to IgG2. So even though the same cell does not produce two isotypes, this is a polyclonal response. And the same individual could have IgMs, IgGs, IgMs, and IgEs, etc. And also through time, the isotype pattern could change. This is idiosyncratic and it needs to be followed by individuals. Ultimately, for antidrug antibodies, the isotype characterization is not as important as understanding that there's presence of antidrug antibodies that could be getting rid of the drug or could be producing safety effects in each person, and they have to be followed on a case-by-case basis. So let's finish with the last three statements. The differentiated TH phenotype drives the specific isotype switch to produce antidrug antibodies of IgM, IgG, IgA, or IgE isotypes. Well, the trick here is that they don't switch to IgM. They always switch from IgM to the other isotypes. But it is the differentiated TH phenotype, the one that drives the switch. Antidrug antibodies can switch isotypes in any individual at any time after administration of the drug, and it is idiosyncratic. Yes, this is true. Drugs as antigens are danger signals that always trigger antidrug antibody isotype switch. This is not true. As danger signals, immunogenicity to a drug can actually happen in the absence of antidrug antibodies, and it can happen in the absence of antidrug antibody isotype switch. So this is false because of those two statements. I hope this was useful, and I hope you tune in to my channel, and if you like it, please subscribe. Thank you very much.